There are more than 300,000 species of flowering plants. We call them angiosperms. And there are dozens of them that have important economic value to people. Hundreds of minor crops and dozens of major crops, also plants for fiber and plants for medicinal purposes. And a lot of our medicines come from flowering plants. Of these hundreds of thousands, only a few have been selected for genome sequencing. Mostly that is the plants that have economic value as crops or medicines or model species that are particularly easy to experiment with. Amborella is special because it represents the earliest branch off of the tree of life of flowering plants. And because of this, we can use it to form a triangle in comparison with important crop plants like the grasses, plants that produce fiber for trees. And we can compare, for example, monocots and dicots by using Amborella, which branched off before the separation of the monocots and dicots. We can use it to understand the differences between them and their similarities, and probably to identify how new genes come about within flowering plants and what kinds of genes make the difference between important lineages of flowering plants that really diversified quickly, like the eudicots and the monocots. It's become possible to sequence a fairly large genome like Amborella's in a relatively short period of time and for much less money than it used to cost. Amborella is what we call about a medium-sized genome. It has about 1 billion bases of DNA sequence in its genome, and our being able to sequence Amborella has been made possible by a new kind of sequencing we call next-generation sequencing that basically is producing sequence data 1,000 times faster and many times cheaper than the more traditional methods. Nearly every crop species we know has a somewhat different set of genes. They're variations on a theme that are common throughout angiosperms, but where there are new biochemical pathways or where there are novel patterns of gene regulation or even new genes, it's important to have a comparative framework for understanding how those genes came about and how they diversified. Amborella will be critical for that comparative framework where we will make it possible through our websites and through the data from Amborella to compare genes that are found in all of the different important, economically important plants, and to target those genes that are novel within specific lineages. Now, because Amborella branched off earlier than all of the other flowering plants, it's also very likely that it will be a source of new genes that may be useful in our studies of other processes in other crop plants and other economically important plants. There are many flowering plants that have been important for producing medicinal products. Digitalis is one of those. Opiates that are found in the poppy family are another. The origin of these biochemical pathways is very important to understand, and understanding how these pathways diversified requires a comparative framework. And so although there's a very good reason to study the genomes and the genes in each economically important plant, we hope to create a comparative framework for understanding where those genes did originate, and Amborella will make it possible to make that comparative framework. So the evidence that Amborella is a very old lineage of flowering plants really comes from comparisons of DNA samples that have been taken from all the different major lineages of flowering plants. We've compared, and other groups have compared, chloroplast DNA mutations, mitochondrial DNA mutations, and nuclear mutations, and found that trees that are built to represent those mutational differences consistently place Amborella at the base of the tree, meaning it's the first branch off of the tree of living angiosperms. Well, this is a project that goes back more than 10 years. We first got going on studying the genetics of Amborella in a project called the Floral Genome Project, which involved scientists now at the University of Florida, SUNY Buffalo, Kew in England, and others at Penn State. And in that project, we started to learn what genes are important in the process of flowering in Amborella. And since that time, we've collected a large amount of sequence data based on the genes that are expressed in Amborella. And we've built resources for being able to efficiently sequence the genome of the plant, made clones and made libraries. And it's taken about this long to reach the point where now we could make a very compelling case that it would be cost efficient and scientifically appropriate to sequence the genome at this time. Flowering plants represent the dominant forms of vegetation in most parts of the land of the earth, and how plants respond to change and how plants will respond to global warming is a critical question facing us now. The potential for finding new genes in Amborella or understanding how genes or gene regulation occurs better throughout the angiosperms will help us deal with these problems in the future. So one of the very interesting questions that will be addressed with the Amborella genome, in addition to the science that we've talked about, is whether or not flowering plants originated through a genome duplication event. We call this polyploidy. And there are different data sets that offer different contrasting evidence that suggests either angiosperms did or maybe did not originate through polyploidy. We feel sure that the Amborella genome will resolve this issue with some degree of confidence.